Marhamat, savollar bo'lsa, ma'ruzachiimizga savollar bo'lsa berishlaringiz mumkin. Yoki ikkinchi varianti, reglamentlar chiqib ketmasligi uchun savollarni yozma ravishda elektron pochtaga yoki umumiy chatimizga yozilsa, albatta bu savollarga tegishlicha to'laqonli ma'lumot ma'ruzachilar tomonidan beriladi. Rahmat. Kun tartibi bo'yicha ikkinchi mavzuga o'tsak, demak, Hindistonning Allahabad universiteti biotexnologiya markazi professori Shanti Sudramga beriladi. Demak, mavzu mavzusi qishloq xo'jaligida biosken sirlarining roli deb nomlanadi.
Menga tayyor.
Marhamat.
There was key moment, it's like a challenge. Due to the limited time, so if there will be any questions, you can chat or directly send an email to the speaker about your questions or comments, and they will answer. And so moderator is inviting a second speaker, Dr. Shuntaram, and her topic will be role of biosensors in agriculture.
Yeah, thank you, Professor. Now I would like to invite Professor Shanti Sundaram. I would like to introduce her first. She's Professor Shanti Sundaram is a gold medalist in microbiology and microbiology from Bombay University and has completed her PhD in microbiology from Barkatullah University, Bhopal. She is a peer member in NAC committees and has been appointed as UGC nominee of the governing body for Shri GVG Visalakshi College for Women in Tamil Nadu. She is invited professor at Second University of Naples, Casata and Italy, and was British Council postdoc at Department of Biological Sciences, University of Warwick, UK. She has been constantly involved in active research since 30 years, publishing more than 150 research papers in high impact factor journals. She has successfully guided 21 PhD scholars. She is a member of some of the prestigious international societies such as American Biophysical Society USA, American Society of Microbiology USA, and Infectious Disease Society of America USA. She has successfully completed 10 national international projects funded by DST, DRDO, DBT, etc. She has attended 125 national and international conferences. So now I would like to invite her to give her presentation. Professor Shanti Sundaram, please come forward and give your presentation.
Thank you, Priyanka. I'll be just sharing the slides first. Thank you.
Professor, I have right now.
Okay.
So after your presentation, you will make me the host.
After the presentation. Okay. Okay, Amit. अरे क्या ये सब क्या आ रहा है? Is it visible, Amit?
Visible. Professor, it is visible. Just click F5 to make it full screen.
F5. I don't know तुम कर. Full screen करिए ना. Cancel. Full screen. ये. Priyanka, shall I start?
Yeah, ma'am. It is visible now. You can start.
Okay. So good morning to all of you. At the onset of the discussion, I would like to thank Dr. Amit Goel and Dr. Priyanka Singh for giving me this opportunity to interact with the erstwhile members of Uzbekistan It's indeed an honor that these two young, faculty are taking this initiative and making the relations healthier on the scientific platform. Both countries, India as well as Uzbekistan, have an agricultural economy, and the GDP is totally based on agricultural economy. So I congratulate both of you and the members of Uzbekistan for organizing such a brilliant platform for interaction. And I hope you grow further and we have further such discussions. So today I'll be talking about the role of biosensors in agriculture. As all of you know that because both Uzbekistan and India, we are an agricultural nation, every small bit of the environmental changes is going to affect the country's economy. There are a lot of negative impacts on farming today because of the high demand that is required for food globally with enhanced population of the countries. Hence, we have to go for smart agriculture, smart farming, and use latest technologies. These latest technologies in the form of smart sensors is going to help the farmers not only in the productivity, to utilize resources which is available in very meager amounts. Environmental changes are vast and they can avoid disasters. What is a biosensor? A biosensor is nothing but an analytical device which is used for detecting an analyte, which I will talk to you in my later slide, that combines a biological component with a physicochemical detector. So as you can see in this present slide, you have an analyte which is attached to a platform. And when the analyte attaches to a connecting molecule on the platform, there is a detection in the form of electrochemical, potentiometric, in the form of a change in the weight, in the current, and so on, which tells you how the binding has taken place. So what are the analytes which you can detect using a biosensor? It could be a protein molecule, it could be a toxin, a peptide, a vitamin, sugar, metal ion, pathogen, and herbicides. How are we going to transduce this attachment in the form of an analytical unit? It could be antigen antibody, it could be a substrate and enzyme, it could be attachment to specific membrane receptors, or it can be competitive binding. Now, this binding will produce a signal. The signal can be of various kinds. It could be an optical signal, an electrical signal, an electromechanical signal, quartz crystal microbalance, thermal magnetic pressure, or a change in the weight or mass. So what is the important role of these sensors in agriculture? They can tell you the changes in any kind of motion due to winds, heat, light, and they also provide the farmers with important data such as any change in the environmental conditions. They can use drones or robots to spray pesticides and insecticides. So what are the different types of sensors that can be used in agriculture? You can have mechanical sensors. You can have optical sensors where light is used for detection. You can have airflow sensors that helps you to see the compaction of the soil, location sensors, moisture sensors, and electrochemical sensors. So here we'll talk about optical sensors, which helps to determine how much clay content is found in the soil, how much of organic matter is there in the soil, what is the moisture content. Massachusetts Institute of Technology has come up with a nano optical sensor that helps to detect arsenic, which is a highly toxic heavy metal found in the soil. Electrochemical sensors are those sensors which help to detect moisture, alteration in the pH, any deficiency in the nutrition, alteration in the temperature, and also the presence of pesticide and herbicide. As you all know, this is being enhanced and used widely by the farmers to prevent pest contamination. Mechanical soil sensors are helpful to see whether the soil is breaking, there is cutting of the soil, there's excess moisture content present in the soils, and the conditions of the soil, whether it is optimum or not for the growth of any crops. In vitro monitoring is essential because in case of in vitro monitoring, if you are traveling and you want to see whether your crop is growing properly, it is required to carry a mobile sensor which helps you to monitor. Soil moisture sensors help to detect the soil content. In case of the rainy or the monsoon seasons, the amount of water present in the soil should not exceed an amount which is not going to help this crop grow and destroy the crop. Location sensors are essential. If the farmer is moving around in the field, he needs to be in contact with GPS satellites to find out the range and distance and the height from whatever position he or she are located. Electronic sensors, they help to convey the data of the exact situation in the field condition to the farmers who may be in a mobile situation, who in turn will inform various units where the field executive is working on the crop growth. Airflow sensors again, these are very useful to see what is the pressure within the soil, what is the compaction nature of the soil, what is the soil type, and what is the structure of the soil. I'm going to, in very brief words, tell you practically I have designed 2 very successful sensors in my own laboratory here in India. And I would like to thank the funding agencies who provided me the funds to design these, that is the Defense Lab of India. I have designed a silica-based immunobiosensor using diatoms to detect fungal pathogen. In India, fungal pathogen of wheat crop was very disastrous at one point of time, and it severely affected the economy of our country. And hence, trying to find out the pathogenic seeds, the seeds containing the spores of the fungi at an early stage would help to prevent such an economic loss. So I tried to use diatoms, which is a kind of algae. Algae are photosynthetic prokaryotic organisms, and diatoms were specifically used because of its 3-dimensional silica-based structure, which provided a large surface area. Diatoms have been used for various applications throughout the world. It is being used in pharmacy, for synthesizing biomedicals, for waste bioremediation, nanotechnology, biosensing, for which I have used in my own sensor, immunodiagnostics, filtration, and drug delivery biomineralization. So in my lab, what we did was we got the diatoms from various river sources, and separately we cultivated the fungal spores and isolated the protein from the fungal spores. So as you saw that it is an immunosensor. So an immunosensor means I am immobilizing my antibody onto a solid surface and waiting for the spores, which is the protein source, which is the antigen source, to bind to the antibody. So in such a way, my cantilever-based platform was designed. and I attached the antibody to the platform, and it can be kept in a field as a portable sensor. And if there are fungal spores present at the wheat stage of the seed stage of the wheat, it can be shown by a change in the weight of the sensor or through a vibrometer where there is a deflection. So that is called as a piezoelectric sensor. We also designed an optical biosensor where this attachment was studied using a photoluminometer. So this is a photoluminometer where the photoluminescence was studied. And this is the basic structure of a biosensor which we had designed. As you can see in this image on the right, the figure on the right is showing you the cantilever beam, the 3D silica layer, the antibody layer, which has been prepared for specific Tilacea, the fungal protein. Now, besides this, the second one which we did was there is extensive use of pesticide and herbicide in the environment to get healthy crops. The farmers don't realize the use of these pesticides, heavy metals, and herbicides is very harmful to the crop and for the fertility of the soil. And hence, we've designed a biosensor where we try to use this aspect of the algae. This is the Z scheme where there are two photosystems present in the algae. The first one, photosystem 2, is able to take electron from the sunlight and splits the water, and water releases O2, oxygen. It is this oxygen that is detected using a Clark's oxygen electrode. When there is herbicide, heavy metal, pesticide present, this oxygen evolution is inhibited, which you can detect using a oxygen electrode. And these are the two successful sensors which we have designed, and which, as Uzbekistan is also an agricultural country, is going to be very useful when it comes into the market. And we are hoping to design many more such sensors that is going to help the agricultural economy of this country. Thank you so much. And I'm very proud to show you this picture, which is the University of Allahabad, where I come from. It is one of the 4 oldest universities of our country, 135 years old. And I'm very proud to be a faculty of this university. And I once again thank the rector, Professor Usmanov, another rector, Zamidov, Dr. Priyanka Singh, and Dr. Amit Kumar Goel for giving me this wonderful opportunity, and hope we keep interacting again. Thank you so much.
Yeah, thank you, ma'am, for sharing such an informative session for the application of biosensors in agriculture. Hope this will be very helpful for the researchers present here. So now I would like to give the mic to Professor Notoji to invite our third speaker. Professor Notoji.
Before that, Priyanka, I ask Professor Shanti ma'am to please make me the host back again.
I have done it, Amit. I've just changed it.
Okay,
Thank you so much.
Thank you, ma'am. Thank you, ma'am. Thank you.
Keyingi ma'ruza navbatini o'simliklar biotexnologiya sohalari, o'simliklar biotexnologiya sohalari mavzusida biologiya fanlari nomzodi dotsent Murakbar Yakubovga beriladi.
Next speaker, Dr. Murakbar Yakubov. His topic is the fields of plant biotechnology.
Dr. Murakbar Yakubov has 20 years of laboratory experience. He obtained his PhD in biotechnology and studied the transformation of tissue culture of cotton by agrobacterial methods, and defended diploma work under the title Integration and Expression Exergenic Information of Cotton Genome. As a researcher, GMO standard, research in food security, plant cell cultivation, suspension culture of plants, induction of somatic embryogenesis from callus culture of plants, is an isolation of DNA, transformation, agrobacterial and ballistic, and regeneration of plants, obtaining and adaptation of transgenic plants. He has presented more than 50 publications, participated on several STCU, UNEP, and FAO projects as researcher, member of Information Management Group, and head of Public Awareness Company. Now he is a head of Plant Biotechnology Laboratory and Research Institute of Plant Genetic Resources, Uzbekistan. Floor is yours, Dr. Mehmet.
Dear guests and participants, let me introduce my topic, field of plant biotechnology. Ethical consideration
Toksuzdur orman ya. Talking good.
needs as much as 70% to support the growing population in the world, about 10 billion in 2050. It is important to consider whether and how genome editing technologies can contribute improving the efficiency of food, feed distribution, and reducing waste. Lower middle income countries face a situation where demand for food, feed, and fiber, climate change, and productivity threats adversely impact their nutrition and livelihood security. Almost 12% of the global population is about 928 million people, was severely food insecure in 2020. To meet the demands of the expanding world population and climate change, it is critical to accelerate plant and animal. The safety of food human consumption is a key concern. There are also concerns about the
Lan sohbet yani var. Sohbet yani. Sohbette. Hadi de.
cell engineering. Cell engineering is a field of biotechnology the conservation of cells and feature on nutrient media. This method has a number of advantages our existing traditional methods as obtaining genetic homogeneous material, release of plant from viruses, high multiplication factor, reducing the duration of the selection process, propagated plants that are difficult to propagate by traditional methods. The possibility of carrying out work through the year. In the middle of the 19th century, Theodore Schwann formulated he summarized the
Institute. Laboratory.
is a
1 minute, Professor.
of all living organisms.
No.
Please mute the mic.
Allah'ım.
And plant are similar in structure.
İngilizce yapıyor değil mi? Ben 35 gereken fizede.
Schwann introduced
Что geçeyim
the
gerek?
inter-
Bu soru iş gerek.
understanding of the independent unit of the life.
Bir sekund.
Professors, please unmute your mic. Please mute your mic.
Bo'ldi.
Endi yo'q ekan. Biz ham yo'q.
O'zimizga yo'q turapsiz yana mana bizga.
Bo'ldi, gapiraver. Gapiraver.
Improvement of plant and animal based on cell technologies. Plant cell and tissue grown on artificial nutrient media form the basic of various technologies in agriculture. Some of them are aimed at obtaining plants identical to the original form. Other are to create plants that are genetically different from the original, either to facilitating and accelerating the traditional breeding process for creating genetic diversity and searching for and selecting genotypes with valuable traits. Genetic engineering. Genetic engineering is based on obtaining hybrid DNA molecules and introducing these molecules into the cell of other organisms, as well as on molecular, biological, immunochemical, and biochemical methods. The use of genetic engineering makes it possible to obtain plants with genes for resistance to disease, herbicides, pests, foliage, during storage, and stress. Transgenic fruits have a longer ripening period than conventional crop. This factor has a great effect during transportation when there is no need to be afraid that the product will override. Genetic engineering can cross tomatoes with potatoes, cucumber with onion, grapes with watermelon. The possibilities here are simply amazing. The size of appetizing, fresh appearance of the resulting products can pleasantly surprise anyone. Genetic engineering began to develop since 1973, when American researchers Stanley Cohen and Emily Chen inserted a bacterial plasmid into the DNA of frog. Then this transformed plasmid was returned to the bacterial cells, which began to synthesize protein, and also the transfer from DNA to their descendants. Thus, a method was found that allowed foreign genes to be inserted into the genome of a certain organism. Genetic engineering finds wide practical application in the sector of the national economy, such as the microbiological industry, the pharmacological industry, the food industry, and agriculture. Stages of obtaining transgenic organisms: the gene isolation, inclusion of the gene into the vector, delivery of the vector to the cell, cultivation of the organism. At present, transgenic forms of plant have already been obtained. Tomato, Mosen 260, soybean, Mosen 200, cotton, Mosen 150, pumpkin planters, more than 80, as well as wheat, sunflower, apple, strawberry, etc. Next. Example of genetic engineering. Indo-Uzbek. Important. The technology of switching of cotton genes is an innovative technology of the 6th generation. It was developed by Uzbek scientists in 2008, and further research with the support of the government of Uzbekistan led to creation of high-yield, early maturing, long-stay, drought and salt-resistant vintage cotton varieties of the Orlok series based on this technology. This technology allowed for the first time in the world to simultaneously improve several agronomical variable traits of cotton, which is the main problem in the selection of its new varieties. As a result, the average yield of new cotton varieties reached up to 40 centers per hectare, which made it possible to obtain high-quality fiber with a microneia of 4.2. a strength of 37.8 and fiber length about 40, etc. Next. Production of seed potatoes of high elite classes based on self-technology. In this slide, showed obtaining tubers from lab to farm. Non-virus tubers give for us high yield about 45 per ton per hectare. On this slide, we showed steps of cell technology. Higher, we see from meristem to plants, and down slides we showed the steps of cell technology, hormonal regulation, where you see stems only potatoes, only tubers obtaining without stems. It's hormonal regulation. So, next. The development technology allows for the short period, about 2-3 years, to completely replace imported potato varieties with local ones. In this slide, we showed a producing of non-virus elite potato seeds. One plant of potato give for us about 20 tubers. Next. Summary. So, plant biotechnology being one of the main directions of scientific and technological progress, actively contributes to the acceleration of the solution of many problems, such as food, agriculture, energy, and environmental problems. Thank you very much for attention.
Kun tartibi bo'yicha reglamentimizdan 2 minut chiqib ketyapmiz, shuning uchun reglamentga e'tibor qilishlaringizni so'radim. Keyingi ma'ruza yuqori samarador suyuqlik xromatografiyasi sifatni nazorat qilishda va mahsulot ishlab chiqarishda kuchli vosita, amaliyot, istiqboldagi tahlillar. Toshkent kimyo texnologiyalik instituti professori Octavio Carloga.
The next speaker is a doctor of Tashkent Chemical Technology Institute, Dr. Octavio Carlo. Title of topic is the HPLC, a focal tool for quality control and product development and overview from manufacturer perspective.
Professor Octavio Carlo has over 15 years of laboratory experience and 5 years of experience in commercial analytical laboratory management, where several analytical techniques were implemented, run, and accredited, including gas, liquid, and ionic chromatography, mass spectrometry, infrared spectrometry, metal analysis by atomic absorption, and inductively coupled plasma, plus full vapor metal analysis, among several others. He has presented 18 publications, including book chapters, research articles, and abstracts of symposiums, plus several lectures and other types of collaboration. He obtained his Doctor of Science degree in plant biotechnology at the Centro de Investigación de Estudios Avanzados del IPM, Chinchislav IPM, in Irapuato, Mexico. And his Bachelor of Science degree in Pharmaceutical Sciences by Institute of Pharmaceutics Alimentos of the University of Havana in Havana, Cuba. Beginning January 2023, Professor Octavio Calvo is working in Tashkent Institute of Chemical Technology.
Good morning. I will give a brief list of HPLC, a powerful tool for quality control and product development. An overview from a practical perspective. Why is it necessary a tool for quality control and product development? The next, please. It doesn't work for some reason. Okay. For manufacturing processes, is for minimizing the risk for patients or consumers, evaluation of impurities, contaminants, and complying with legal requirements. And they go by hand in hand with the other ones because on legal requirements, there are some specification of the amounts of contaminant that should be present at any time. Also for labeling, among many other different requirements. For product development, it's for isolation of active principles or ingredients, characterizations of the different ingredients, evaluation of products, structure, elucidation, among many others. One of the main, because of how widely used tools are available right now, is chromatography. Chromatography is a technique for separation, and it's based on the different cell interaction of different compounds in a sample, while they're being carried out through our system, and the carry is called mobile phase, while there is another phase which is called the stationary phase. It is called chromatography because originally it was developed for separating a mixture of colors, hence its suffix chroma. In this small example that we have in here, there is originally the mixture, and they will be run through the chromatographic system. The red compound in this particular case will have the highest affinity for the mobile phase, whereas the lowest affinity for the stationary phase. Therefore, it will be the one who will be dragged out first. Therefore, the retention time, which is the amount of time that it will take for an analyte to go through the whole system, is the shortest one. The second compound, the blue one, is the second one, and the third one will be the last to review because its affinity will be the highest with the stationary phase. Therefore, it will try to stick within the system. Chromatography is generally classified according to its mobile phase. In this particular case, since it is liquid, it is therefore liquid chromatography. There are many other types of chromatography, such as gas chromatography, thin layer chromatography, among many others. High performance liquid chromatography, which is the one that concerns us here, is a liquid chromatography where the solvent is delivered through high performance. Originally, it was high pressure, and the heat meant pressure pumps, which allow, and that's the most important part of it, for a stable solvent flow rate, as well as in many cases for solvent gradients that will be used as a mobile phase. My colleague already, Dr. Ismail already explained how the HPL system works, and he went through a lot of different types of analysis and application. I will very briefly describe some of the conditions required for separation, so as to be able to illustrate my main point here, which comes to product development and optimization. Therefore, the same equipment that Dr. Ishimo showed, I made just a schematic representation of it. It has the solvent repositories, the gasser, pumps, injector, column where the separation will take place, the detector where the signal will come out, will be registered by the computer. Something very important to consider in the case of HPLC, since the type of separation is given mainly by the coefficient of partition, there may be some other cases, but since those two, those are very important driving factors, there are two different types, which one of them is the normal phase, where the stationary phase is polar, and the mobile phase is non-polar, whereas in reverse phase is the other way around. The stationary phase is non-polar and the mobile phase is polar. There are some other types of chromatography, liquid chromatography, which is a